Without further ado, I am delighted to introduce two trainee speakers who are co-first authors on their paper that they are study that they are presenting here. And the first, or not the first, but one of the speakers is Agnieszka Pancholi. She did a Bachelor's of Science in Molecular Life Science in Lübeck and a Master's of Science in Biochemistry and Biophysics in Freiburg. And she did her internship and master's thesis in Leonard Hilbert's lab, where she is presenting her work here. And now she's a PhD student in Sylvia Earhart's lab. Welcome. And then our other trainee speaker from today is Tim Klingberg. He studied integrated life sciences and got his Bachelor's of Science at the Friedrich Alexander Universität Erlangen-Nuremberg, and then studied integrated life sciences for a master's at the same place. And now he is a PhD student in mathematics and life sciences and at Nuremberg. And we are really excited to hear his seminar today. And he is from the Zaberdayev group. So we have two research groups represented here. And thank you so much, both Tim and Agnieszka, for presenting a dual talk. I'm really excited. And for those of you in the audience thinking about if you're co-first authors, feel free to also do co-talk. We're really excited to support that. And yeah, thanks so much. And I'll see you guys after your talk.
Thank you very much for the introduction and for giving us the opportunity to present our work here today. So today we would like to talk about RNA polymerase II clusters and their formation. So it is long known that transcription occurs in hubs called transcription factories, and they consist of RNA polymerase II, coactivators, transcription factors, and others. But today we will talk about RNA polymerase II. As you can see on the second image from the left, RNA polymerase II builds clusters that have different shapes and sizes. So how are they formed? When a gene is going to be transcribed, RNA polymerase II is recruited to the proximity of the promoter and it engages it. Then it can transcribe few base pairs and it becomes paused. Both initiated and post Pol2 can detach from DNA, and all those Pol2 molecules become a part of macromolecular cluster. After pause release, RNA polymerase II it can enter the gene body and it can stably transcribe the gene. Due to the production of RNA, elongating Pol2 is excluded from the cluster. So how can we know if given Pol2 is in recruited or in elongating state? We can do it using C-terminal domain of RPB1 subunit of Pol2. Recruited Pol2 has serine 5 residue of the CTD domain phosphorylated, and elongating Pol2 has serine 2 residue phosphorylated, and serine 5 phosphorylation mark is abolished. This means that we can distinguish between elongating and recruited Pol2 depending on the phosphorylation pattern of the CTD domain. And in our research, we wanted to answer the question, how are RNA polymerase II recruitment and elongation related to cluster morphology? To answer this question, we performed live imaging of zebrafish embryos, and we stained serine 5 phos as a mark for recruited Pol2 and serine 2 phos as a mark for elongating Pol2. Then we processed the images, and we were able to divide the clusters by eye into 3 groups. One of them is small clusters, type 2 is big round clusters, and type 3 are big dispersed clusters. Type 3 clusters we also could had also undertypes. For example, donuts, these are round clusters with a hole in the middle, croissants, which are crescent shapes, or intertwined jalebis. To quantify the clusters in more details, we used STAT-D super resolution microscopy. And we got serine 5 phos channel in super resolution and serine 2 phos channel in confocal. And again, we were able to divide the clusters into 3 groups by eye. To quantify the clusters in more details, we prepared a pipeline which segmented the clusters and calculated 2 values for each of them, area and solidity. Solidity is a measure of roundness of a given object and can be calculated by dividing area by the convex area. Solidity has a value between 0 and 1. Perfectly round objects have solidity of 1, and the more dispersed the object, the lower the solidity. So then we plotted solidity against area for all the clusters, and we gated the extreme cases for all the 3 types that we were able to see by eye. So for type 1, we would have the small clusters with low area. For type 2, we would have big clusters with big area and high solidity. And for the type 3, we would have high area and low solidity. Then we plotted the serine 5-phos and serine 2-phos intensities for all the clusters. And we put the median values for all the gates. And so you can say that the type 1 clusters have low serine 5-phos and low serine 2-phos intensities. Type 2 have high serine 5-phos and high serine 2-phos intensities. And type 3 has intermediate serine 5-phos and high serine 2-phos intensities. This means that different morphologies of the clusters depend on the elongation and recruited Pol2 levels. But we also noticed, you can see this on the right on the graph, that the elongating Pol2 builds foci at the borders of the recruited Pol2 clusters. And previous papers suggested that the clustering of recruited Pol2 can be explained using liquid-liquid phase separation. 2 main hallmarks of liquid-liquid phase separation are Ostwald ripening, where larger clusters grow due to the dissolution of smaller clusters, and droplet fusion, where 2 droplets merge into a single droplet. So we wanted to know if Pol2 clusters exhibit those 2 hallmarks, and we used live imaging for that. So when it comes to Ostwald ripening, we could see that over tens of minutes, the clusters move, but they do not change their main shape. And the growth of larger clusters due to the dissolution of smaller clusters is not visible. When it comes to droplet fusion, we could also see that 2 droplets merge, but they also disconnect over time, which means that there is no droplet fusion. This would suggest that liquid-liquid phase separation alone cannot explain RNA polymerase II cluster formation.
So based on the observations in the wet lab and also other previous work, we then asked the question which physical mechanism and also which species contribute to this process of cluster formation. So we then came up with the following hypothetical model that overall involves 4 different species. So the first species, shown in red, is liquid material containing RNA polymerase II, so the recruited status. And this is the material to form the clusters. In order to form clusters, we assume that this species has self-affinity. In addition to the red species, we also have chromatin that serves as a surface for cluster formation. And this chromatin has 3 subregions. The first one is inactive chromatin, shown in black, for which we assume self-affinity. Then in blue, we have regulatory chromatin, which has an affinity towards the red species. And directly adjacent to this blue region, we have active chromatin, which represents the effect of transcription elongation. And since, as Aga said, elongating polymerase II is excluded from these clusters, we assume repulsion of gray to red. This hypothetical model we now want to represent on a 2-dimensional coarse-grained lattice. So therefore, we use a lattice. Polymerase II we represent as single lattice sites, and our chromatin we represent as a polymer, so as connected lattice sites. We also represent the different subregions of the chromatin with the respective colors black, blue, and gray. And in case a polymerase is sitting on top of the polymer, we just mark it by a cross with the respective color. From this static picture of our model, we now want to add dynamics to it. So for the polymerases, we assume that it can move to all of its 8 nearest neighbors. And what you can see here on the left is the movement of a single polymerase within a lattice of size 25 by 25. And you see the movement in every step. And on the right side, you see the movement of overall 100 polymerases in a fast forward fashion. Also important to mention in here is that till now, we don't include any affinities, and the borders of the lattice are just strict borders. For the polymer, we use the well-known Verdier-Stockmeyer move set that consists of these 3 move types and which has been used to simulate self-avoiding polymers. So also for the polymers, we can have a look at the movement of a single polymer in every step. And here it's a polymer of overall length of 20, and it's shown the movement of in every step. And on the right, we see the movement of overall 4 polymers within the lattice, but again in a fast-forward fashion. Now we want to pay attention to the added affinities to our model. So as I've mentioned them previously. So at first, let's have a look at the affinity of the red species. For a pretty low affinity parameter, the polymerases are just randomly distributed within the lattice. If we increase the affinity parameter by a bit, we see that a little bit of accumulation of red particles occurs, but no real clustering happens. And if we go even higher with the parameter, then we see that phase separation on its own happens. But now the question arises, what happens in case we add a polymer to it, as described previously? So what we do is we add a polymer to the simulation of length 20 with, again, a blue subregion. And the subregion you can see here marked by the blue crosses. And in case of the low affinity parameter for red, we just have the coverage of the blue region. In case we again go a bit higher with the affinity parameter, really cluster formation on top of the regulatory region occurs. And in case of the even higher affinity parameter, unsurprisingly, we again just have phase separation. So the mid parameter set is the one which is closest to our experimental results. So therefore, this is the one we will use in the further course. And for better visualization, you here again see fast forward movies of the simulations with and without a polymer. So overall, we can say that cluster formation is in line with surface condensation. And surface condensation has been shown in vitro in recent publications, and we'll now also see it in vivo in the zebrafish embryo. So in the last step, we now add also the mentioned gray species to our model. And what we then did is we did simulations of 4 polymers with random amount of blue and gray subregions. And in here, you'll see an exemplary output of our simulation. But then the question arises, how can we compare these lattice outputs to our experiments in the wet lab? And also, how is it possible that we can use the same image analysis pipeline? To do so, we produce synthetic microscopy images by just adding a Gaussian filter to the lattice output and also adding phosphor noise. And then we can represent in magenta the recruited polymerase and in green the elongating polymerase. Then performing multiple replicates of our simulation, we again get different cluster types. And again, we got, which are shown in here in these exemplary images. And these cluster types, which we could already categorize by eye, we again also wanted to quantify. So we again examined our clusters according to the solidity and the area. And then also we were able to do the same gating as Aga explained previously. We then plotted these clusters according to the intensity of serine 5-phos and serine 2-phos, which is the gray cloud. And on top of this gray cloud, you see the median for each gate. And also in here, we see differences for each gate or each cluster type. So by this, we can say that our model is able to reproduce the experimental results.
We wanted to know if our model reacts to perturbations in the same way as cells. So we treated pluripotent zebrafish stem cells with the inhibitors, either with flavopyridol or with triptolide. And we compared serine 5-phos and serine 2-phos intensities to the untreated cells. So flavopyridol blocks RNA polymerase II pause release and elongation. And so the serine 2-phos levels are lower in this kind of cells. When it comes to triptolide, it inhibits recruitment and degrades POL2 through hyperphosphorylation of serine 5. So again, we have lower serine 2-phos levels and lower serine 5-phos levels inside of the clusters, but overall serine 5 levels in the whole nucleus are higher due to the hyperphosphorylation of serine 5. On the right side, you can also see the analysis.
Okay, so then we ask if we can capture the same inhibitor effect with our model. So here on the left, you again can see the exemplary images for the lattice output and also for the synthetic microscopy images. And for control, we just use the usual parameter set, which I described previously. For flavopiridol and the inhibited pause release, we just removed the gray ends, which represented the effect of transcription elongation. And for triptolide and the inhibited recruitment of polymerases, we reduced the red-red and red-blue affinity. Okay, so then these occurring clusters, which you can see in the exemplary images, we then also wanted to quantify again. And the first measure we used was the solidity of each cluster. So we can see that for flavopiridol, overall the solidity increased, whereas for triptolide, the solidity of the emerging clusters decreased. And if we now have a closer look on the emerging number of clusters per lattice, we also see that for flavopiridol, we mostly have only one cluster within the lattice simulation, whereas for triptolide, we have 2 or even more clusters occurring. And now if you compare these simulation results to the experimental results, we can say that also in here we have the same qualitative trend. So by this, we also can see that we at least can capture these experimental results. So with the graphical summary, I'll finish our talk now. So overall, what we observed is the formation of macromolecular clusters containing recruited RNA polymerase II at regulatory chromatin. What's also important here is that the elongating polymerase is excluded from these clusters. Within the zebrafish embryo, we then saw clusters of different types, and these different cluster types, as Aga mentioned, depend on the amount of recruited and elongating polymerase II. For the biophysical description, we then used the lattice kinetic Monte Carlo model, and this model was able to reproduce our experimental results and also showed us that our cluster formation is in line with surface condensation. And finally, to induce changes in the cluster shapes, we used the inhibitors flavopiridol and triptolide, and also these results we could reproduce with our model. So now we want to thank all the involved people within the labs and also our supervisors, and especially Leonard for the really nice coordination. And then here is our publication. So if you're interested even more into our work, you can check this out. And now we are happy to take any questions, and we will also stay for the coffee break or discussion afterwards.
Great job, both of you. That was fantastic. And I was very impressed with how streamlined you were between the two speakers. That was really great. Take notes, everyone. Anyway, I did have one question so far, or I'll ask one question. I do have a couple. One question also is wondering, are you able to somehow estimate how many Pol2 molecules are in each of the clusters?
This is a very tricky question because
Yeah,
the C-terminal domain, it has 52 repeats of this serine 2 and serine 5. So I think with this method, we really can't do it. We can just assume that the small clusters most probably have 1 or maybe 2 molecules, and the bigger clusters have more of them.
Mm-hmm. Cool. I have more questions, but before I ask them, if you all in the audience have questions, please feel free to write them in the Q&A box or to raise your hand. Oh, it actually looks like Alina Tarsalainen. If your hand is raised, so I can allow you to talk if you would like to ask your question. So if you should be able to ask your question now. Okay, I'll give them a minute. I had another question. So like, for example, like does the CTD length have anything, like I actually don't know a lot about condensation and phase separation, but like yeast, for example, has like 26, I think, in the 26 repeats. So has anyone like tried like truncating the CTD and seeing any differences in these clusterings?
They did this in yeast, as you said.
Oh, okay. Cool.
And the truncating of this CTD domain led to disappearing of the clusters.
Gotcha. So it's really driven by that, because I was going to ask also like if any of the CTD binding proteins might also be helping driving this clustering.
Yeah, definitely.
Cool. Okay, it looks like Alina might have put her hand down, so I will keep that going. Let me just double check there's other questions. Oh, Benjamin Sabari. Okay, now the questions are starting to come in.
Hi, can you all hear me?
Hi, welcome. Thanks for your
Yeah,
question.
great talk. I really enjoyed your paper as a preprint and as reading the published version, and cool data. Your model is really intriguing in the surface condensation, as you say, it matches previously published data. So in your model, or you're thinking about this, is the DNA or the chromatin around which this forms an obligate or required component of the cluster, or could these form anywhere in the nucleoplasm?
So at least like from our model, I would say that the regulatory chromatin in our case is necessary for the cluster formation.
And do you have any thinking on why specific regions of chromatin would nucleate these or other regions not?
So I would say, so we have additional data which we haven't shown in here, is where we mark the super enhancers, and in there, so we are at the moment thinking about like there's any specific visiting for genes necessary to these super enhancers. So yeah.
Cool. Yeah, I look forward to seeing that.
Cool. And we have a couple questions in the chat or Q&A box. The first one is from Omer Galulu, and they ask, according to the conclusion about the LLPS association, dissociation dynamics of RNA pol with and without RNA by considering spacer sticker model, according to the conclusion. Actually, trying to figure out what the question is there. Omer, if you would like to clarify your question in the Q&A. Okay, thank you. Sorry for that. Okay, so have you considered spacer sticker models and or LARC's RAC's system in the focus of RNA polymerase? I'm not sure about that, so I'm gonna let you, Tim, if you would like to read the Q&A.
Yeah. So
it sounds very
I'm reading at the moment, but we have not further thought about like this sticker and spacer model.
Cool. Thanks, Omer, for the question.
To answer it shortly.
Cool. And so I will take, I think, one more question before we move on to the next speaker. Okay, so Sumitaba, I want to read the question before I ask it, unlike before what I just did. Thanks. Okay. Sumitaba Brahmachari asks, thanks for a nice talk. I agree. Looks like the elongating pol2 are also clustered among themselves, the green blobs. Do they also form a separate condensate?
They form very small foci, so I don't think that you can call it clusters, but they form this small foci at the borders of the clusters. I don't really know if it is condensate. I would assume so, yes.
Cool. And then I think, do we have more time? Okay, there's one more quick question from Nina Ripen. Do you think nascent RNA plays a big role in recruited condensates here, or is it more the chromatin?
That's a very good question. I think it might be both, but we didn't investigate this. So
future studies.
Mm-hmm.
Great.